So, had the projector been working last week, this is a figure I would have shown you, which is very small to you that are looking at it in person, but this is the development of the dog. So early embryonic, mid-embryonic, late embryonic. Next to it is a bat. So the embryonic stages of a bat: early development, mid-development, later development. You can start to see its arms, wings lengthening, and its fingers, wings extending. And then a rabbit in the third position, and human in the fourth. And the point, to reinforce what I mentioned last week and to reiterate, is that early development looks very similar among mammals. It's only later in development when the pathways, the trajectories of how different tissues are growing change. And again, that's the expectation that we come into, sort of the perspective of a developmental biologist, is that we expect early developmental processes to remain the same, and that changes in development will be made late in development. So when you notice any differences between any two closely related organisms, like sister species, closely related species, horses and donkeys, the differences you see are likely results of changes in development. This is my more favorite version of that slide. We expect conservation. So in developmental biology, you, in theory, predict that dogs and humans would look the same, except that there are these late development changes that cause dogs to be different from humans: different stature, lack of opposable thumbs, those sorts of things. To put yet another way, there's this really weird, and this is why Evo-Devo I think is a really interesting field to work in, because it's almost like you're trying to combine two opposites. So evolution is a field that is it's about genetics, because the genes encode the instructions for building organisms. And if you're going to have an evolutionary change in an organism, that means you're going to be tinkering most often with its cookbook, the DNA, its genome. From an evolutionary perspective, most evolutionary biologists think about mutations as, well, that creates a novel organism that's got a mutation. It's a mutant, it's different than everybody else. And in evolution, that mutation is either good, as we discussed last week, based on the environment. If you're the black polar bear cub that I totally made up and doesn't make any sense at all, then that mutation is probably not useful if you still live in the Arctic, but it might be camouflaging if the Arctic ice sheets melt and there's bear water, which there isn't in the Arctic, but that's another reason it was a bad example. So mutations could be good in some circumstances, bad in other circumstances. And evolutionary biologists look at the benefit of mutations as generating diversity. So I was trained as an evolutionary biologist, and then I became a developmental biologist later, so I think I still have those early developmental, academically developmental biases. That mutations are good, except for when they're not, because they create diversity, and diversity is great. Development, on the other hand, as you may be starting to understand, sort of takes the opposite perspective, which is that the goal of evolution is to create diversity and then weed out the variants that aren't useful. In development, it's almost the opposite perspective, that mutations are going to be bad most of the time. Mutation often leads to defects. And it's very rare and this is just a bias, this is not true, this is just the way that different scientists might look at the same question. Evolutionary biologists might say, ooh, diversity, that's great. And the developmental biologists might say, ooh, mutation, that's probably going to be bad for development. And so marrying those two, trying to figure out which is true, is the central purpose of the field of Evo-Devo. Is this mutation useful, detrimental, and as we discussed last week, questions like, when in development does a mutation affect development? If you see the same trait in two unrelated individuals, was that because of the same mutation, or are there multiple ways to get mutations to cause the same trait? So we're going to start this semester with talking about Hox genes. These are an extremely well set of genes, DNA sequences that encode proteins. and their role in patterning. So today is all about background in both, so introduction into genetics, evolution, and development, really brief, but to hopefully bring everybody up to somewhat of a common level of understanding. Then we'll talk about the Hox genes, and that's because this paper, to understand, you will probably want to know something about the Hox genes before you start reading. So before I actually get into the real nitty-gritty, are there any concerns I can address at this point? Questions about anything? Because now we're going to get sciencing. Class. The next full moon. Okay. Oh, yeah. Does anybody have anything to add to our list of, or to begin, rather, our list of organisms that are useful for studying evolutionary development? I'll write them down if you'll tell me what you got.
Sea urchin.
Let's do one each to start anyway. So sea urchin. Okay. Sea urchins they're multicellular, they do develop. Okay. Then there are a lot of studies of Evo-Devo and sea urchins. Did you?
I'm not going to do the Latin name, the common fruit fly.
Fruit fly. Drosophila melanogaster, yeah.
Yeah.
That's okay, we can stray away from scientific names as much as we need to, there's no reason. Okay, so fruit flies. Okay.
Demo sponge?
Which one?
Medium Queensland.
So some sort of sponge.
Yeah.
Okay, cool. You know, like loofahs. Except the real ones that live in the water. Yeah. Bob, do you have one?
A plant, a Arabidopsis.
Okay.
Thaliana.
Yes, Arabidopsis. Thale cress. Yeah.
Octopus.
Cool. What would be interesting about studying development in an octopus?
I saw an article about arm and hand formation.
Yeah, exactly, right? A lot of animals have 2 or 4, mostly 4. So, yeah, how does 8 or 10 or 12 come into being? Very good. And by the way, if somebody didn't do it, don't worry about it. It's not a big deal. Do you have one?
Yes. I know how to say this, so I'm going to spell it out.
That's okay.
Cichlids.
Oh, yeah. Cichlids.
Okay, cichlids. I was going to say like chiclets or something.
Yeah. Back in my day, chiclets were little pieces of gum from chicle, you know. But no, these are, yeah, no, you're absolutely right. A lot of people do call them cichlids, but they're cichlids. They're fish from different lakes in Africa. Yeah, we should do a paper on cichlids. Oh, well, not enough weeks.
C. elegans.
C. elegans, all right. Worms. And,
I have mouse.
okay,
Mice.
yep, that's what we're going to be talking about next week. Vertebrate development. Do you have one, Edgar?
I'm going to say Hydra vulgaris.
Nice. Okay, so we'll put that next to octopus. Hydra. And,
Pristioncus pacificus?
oh, cool. Okay. We've got the nematologist and the Pristioncus. Which, if you have time, I know you don't, but if you want to make time, go to YouTube and look at the video of Pristioncus eating C. elegans. Pristioncus is a worm that has teeth. That they're predators of other worms, and so it can get kind of gruesome for the C. elegans. So those of us that love C. elegans don't like to watch it. It's like a slasher movie for C. elegans. Okay, so what's common, if you know anything about any of these species? So we've got, let's see, we've got insects, fruit flies, C. elegans. We've got plants. We've got invertebrates, we've got vertebrates. Do we pretty much cover the spectrum of living things that do development, multicellular things, in other words?
I've got a moss, Psychometrella.
Ooh, moss, okay. Nice. Xenopus, fish.
It's a
Oh, it's not
frog.
Xenopus, fish, frog, yeah, Xenopus. The African clod frog? What now?
Comb jelly? It's like
Comb jelly. Okay. I think I can only fit one more if somebody wants to add one more thing in here. Zebrafish? Okay. Zebrafish. Danio Ririo, for those of you that are keeping score. Okay. Why do people study development in these organisms? Aside from the fact that they are multicellular and so they do development. So octopus, I think, may be clear. It's got a different number of, and it's the same for Hydra. It's got some interesting development that it does in terms of development of appendages, let's say.
the genomes are known, so
Okay, so genomes, so development is all about how genes instruct the body to develop. So you have to know something about DNA if you're going to study development, and also about evolution. So of all of these, which ones do we know the genomes of? I don't know certainly all of these. Okay, fruit fly and C. elegans, definitely. Mouse, definitely. Arabidopsis, definitely. So that's a fruit fly, a worm, the mouse, a plant. Zebrafish, definitely. I'm going to guess, I know some cichlid genomes are known. I'm going to do these ones in blue then. Pristioncus's genome, Xenopus, probably.
The sponge one is known.
I'm guessing, yeah, most of the sponge Hydra is probably known. I don't know about comb jelly octopus moss. That one might be known. I don't know. Anyway, so yes, that's one category. So if it's something that if it's an organism that we have some genetic information about, that's helpful because we need to know how its genes function in order to create development. Other aspects of these that are useful. What makes working with some of these more or less challenging, perhaps? There are some things that some of these have in common, but not all of them.
A short life cycle.
Okay, so short life cycle, that would be,
Many offspring.
yeah, fruit fly, right, and many offspring. So the characteristics of what geneticists call model organisms. So you want organisms that are small, ideally, so you can grow a lot of them. What else?
Small sequence genomes.
Right, so small sequence genomes, small size, have lots of offspring so that one pair of parents can create lots of kids to study. Physically small, I already said that, physically small. Cheap to grow. Okay, so which of these are not probably cheap to grow or access, or maybe you can't even grow in the laboratory? So let's see, yeah, I'm gonna say, yeah, octopus, yeah, I mean, you can grow them in aquaria, but if you want to study their development, anyway, yeah. Mosses you can grow. Arabidopsis is a fantastic grower in terms of plant development. That's why it's one of the powerhouses in the plant world. Sea urchins may or may not be easy to do husbandry for in the laboratory. I'm not sure. I've never worked with sea urchins. I've eaten them. They taste good. So some of these are better than others in terms of genetics. Size of their genome, whether it's sequenced, and then moreover, it's usually how well they grow in the laboratory. But that's the bench scientists talking. Then there are the more ecology-loving scientists, which I am sort of, but not as much on the spectrum, who love to go out in nature and look at development. And that's why the other half of the organisms are here, because they're cool, they're interesting, they're intriguing, they're compelling. When you want to write a grant application, maybe it's a little sexier of an organism than I want to study a microscopic worm. So, or maybe it's, like a lot of aquatic creatures, maybe more likely to be experiencing rapid climate change, and therefore we want to know something more about how they develop so that we know if changing ocean temperatures might negatively affect their development in the future, right? So to argue for policy. So you get this sort of balance between they're good in the laboratory and they're interesting to study in nature, or they're compelling to study because humans care more about them than just sort of lab rats. So hopefully, maybe, when we discuss all of our papers, we'll talk about, as part of our discussion, what made that creature, whatever it is, amenable to studying evolution and development. So two mammals, both have similar structures, but they didn't develop the same way. So that's a question we'll be talking about, the origin of novelty. And to get there, we have to do a quick series of background slides. Development, study of genetics and how it affects the growth of an organism. So in order to understand that, you have to know that DNA, the double helix, the information that's inside all of our cells, gets read in our cells. That's just the information. That's not, the DNA is not the stuff that does the work of the cells. So DNA encodes a single-stranded nucleotide, so same type of molecule, RNA, that also carries information. The DNA is sort of the stable, it's the book on the shelf, it's always there when we need it. I don't know what the good analogy then is for RNA because it's used briefly in concert with other proteins, in this case the ribosome, don't remember that, it's not important particularly at this point, to produce, so that's the ribosome, reads RNA, this messenger molecule, and produces protein. So that's the central dogma of biology. The DNA encodes the information, and that protein then reads the RNA and creates a protein, which then folds up into some three-dimensional shape on its own inside the cell, and then those proteins do stuff, like DNA polymerase. So some gene in our DNA encodes the protein that's responsible for copying DNA, for making more copies of DNA to pass on to cells as cell division occurs. Other examples of proteins in our cells? We're going to talk about a lot of genes and proteins. Every protein is encoded by a gene, one protein, sort of the old, not quite true anymore, but used to be the mantra. In the DNA, there's one gene that encodes every protein. What proteins help our muscles contract and expand? Yeah, so actin is a type of protein. Myosin is a type of protein that helps when we flex our muscles. That's what causes the cells to expand and contract so that our arms can actually move. It's kind of important for development. Myosin. DNA polymerase. And so we're going to look at, in part this semester, we'll look at mutations in the DNA and how that changes the code for the protein that's produced. So changes in DNA will often produce changes in protein sequence, and therefore how well that protein works. And of course, this is really simplified, but I just wanted to remind everybody this is framing evolutionary developmental biology. Mutations and their effects on proteins is kind of the first layer that we look at. And from there, then, the effects of proteins on an organism. All right. So one of those proteins could be a transcription factor. There are lots of transcription factors that exist. So these are genes that are encoded in our DNA, and their job is to turn on the production of more proteins. So they're messenger proteins, basically. And in this case, this figure is just diagramming one. They're just called generically receptor protein. And it, by itself, just hangs out inside the cell and doesn't do anything. Until, for example, a neighboring cell, or the paracrine or endocrine system, releases a diffusible molecule, like a hormone, that enters the cell. And if that hormone binds to this particular receptor protein, that allows that protein to go into the nucleus, bind to DNA, and now that protein is instructing the nucleus, or reading the DNA, to make a different type of protein, read a different gene to produce a different protein. That's what's going on down here. So transcription factors, also known as enhancers sometimes, are one of the gene families, types of genes that we're going to be looking at a lot Because these genes control whether or not other genes get turned on or turned off. And we'll see why that's potentially useful in a minute, in a sort of abstract, general way, not an abstract way. So when these transcription factor proteins bind to DNA, they do so specifically. So one transcription factor protein will have a specific nucleotide sequence, like ATGT, CTA, or whatever, each of those 4 letters standing for the 4 different nucleotides that make up DNA. The letters of the book of life, in other words. Each transcription factor has a specific DNA sequence it will bind to, which means that each transcription factor only goes to specific parts of specific chromosomes when it's in the nucleus, and so it will either turn those genes on or off, depending on what that protein was designed, for lack of a better word, to do. So Hox genes, we're talking about next week, are transcription factors. Proteins that turn on expression make the DNA read other genes. One example of a transcription factor, specifically how it works, because I thought that was kind of abstract. So transcription factors somehow know how to go to a specific spot in DNA and bind there. I could spend an entire semester talking about how exactly that process works. How does a protein doesn't have a brain? How does it know which part of a chromosome it's supposed to bind to, which DNA sequence it recognizes? And as part of my undergraduate research long ago, I actually worked on one transcription factor, which some of you heard me talk about before. It's TATA binding protein. This is a protein that's present in all of our cells at pretty high concentration, because every time a gene gets read, transcribed for biologists, every time a gene is going to be read and protein production is going to start for that gene, this protein, TATA binding protein, is the first protein that binds to DNA. And so this is a crystal structure of that protein. And it's hard to see. There are some spirals in here. Those are alpha helices for those of you that have had biochemistry before. And there are these stirrups. This has been referred to as a saddle-shaped molecule that sits on top of DNA. And there's these loops, alpha helices, and there are also stirrups on the bottom of the saddle. And the way that TATA binding protein knows which DNA sequence to bind is all about the physical, chemical contacts between the amino acids that make up that part of that protein and the amino acids that are in some of these helices, how they actually specifically interact with different sequences of DNA. So here's another shot of TBP binding DNA. And there are a bunch of, for those of you that like chemistry, there are a bunch of weak interactions made by hydrogen bonding between each of the nucleotides and amino acids that are made up, that comprise those helices, and the stirrups too. So there are all of these individually pretty weak chemical bonds that exist between TATA binding protein and the double helix, but all together, those individual weak interactions add up to a strong force that once, this is the critical part, once TATA binding protein happens to, as it's diffusing through the cell in the nucleus, randomly bumping into stuff, when it gets near that DNA sequence, if it makes all these hydrogen bonds, it'll stick there for a while. And then at some point it'll fall off again. So proteins do recognize specific DNA sequences. It's all about the actual amino acids that make up the different parts of the protein. So lots of proteins have lots of different DNA sequences that they will so-called recognize. And when TATA binding protein binds to a part of DNA, then that gene gets transcribed.
So is the TATA binding protein like attracted?
So here's one. So let's see. So one gene, one transcription factor turns on a set of genes. So it's kind of worthless if one transcription factor, if one protein only recognizes one DNA sequence that exists on one chromosome and only turns on one gene. What these transcription factors like HOX1 do is they bind to lots of spots in the DNA. So let's say HOX1 binds to a 6-letter long sequence, ACTGAT. Don't even have to remember it. I'm just making up a 6-nucleotide sequence. That sequence is going to be located at lots of different spots on lots of different chromosomes. And so when HOX1 is the protein, when it's present in the nucleus, it can bind all of those spots and turn on all of those genes. So one protein, one transcription factor, can make a pretty large effect on what other genes in that cell are turned on, what other proteins that cell produces. So it just happens that HOX1 is actually a gene that specifies in eukaryotes, cells with or organisms with nucleated cells. It specifies where it's turned on, where this transcription factor is active, where the protein is produced, that tells those cells to become anterior, head, this end of the body, not that end of the body. And so it may then turn on, as it's in the nucleus, turning on transcription of other proteins, turn on 2 more, say, transcription factors these are not real, of course, I made them up. Well, actually, not of course. I didn't make these names up, though. I1 and EAR1, I just want to make it obvious. So if HOX1 is in the anterior, if it's expressed, if this is a multicellular organism that's developing, still young, hundreds of cells, thousands of cells maybe, make up this organism. HOX1 is expressed at the anterior, so this end is going to be the head. Well, of course, in the head there are multiple different organs. There are lots of different structures that have to be formed by the time this organism is an adult. And so one of those is, well, you need eyes to form, and you also need, in this hypothetical example, ears to form. So it's a typical case, and this is just hypothetical as it says, that HOX will go around turning on multiple different types of genes, including I1 and EAR1. And it does so in this case, different parts of the developing anterior of the organism. The key point to note here is you can't turn on all of the genes everywhere, you being the cell. If you turn on all of your genes at once, what happens? You've got cells, you've got proteins that are trying to turn your cell into a leg, and a cervix, and a clavicle, and eyes, and brain, and so forth, hair, epithelium. So you have to have discrete areas of gene expression, of genes and proteins being turned on and off that are defined. And that's where these inhibitory relationships come in. So straight arrows in this class and in biology and genetics in particular indicate a positive relationship. So HOX1, the protein in the cell, goes into the nucleus and activates transcription or production of I1 proteins. And it also does the same thing for EAR1. So HOX has a positive effect on these proteins. It turns them on. But it turns out, and this will be true, and we'll see this some through this semester, that then proteins, well, they intersect here, right? Their expression patterns overlap. Some of these cells right here have both the eye protein, you telling the cells they should become eyes, but they're also getting some expression of proteins that are telling them they should become ears. And we'll see this negative relationship between the two where if a cell makes more of the I1 protein, that will repress this blunt arrow, it'll turn off EAR1 production. So there's this balance, which is often shown as a scale. So if you have even the tiniest bit more production of I1 protein, it sort of wins. It can turn off the production of EAR1 because there's more of it. So it wins, and then you get production of I. And in other areas of the body, then the opposite will occur, where these cells might produce a little bit more of the EAR1 protein, so you will eventually get a clear boundary between what's going to turn into an eye and what's going to turn into an ear in terms of cell by cell making this decision of which one is being produced more, I1 or E1, and establishing a clear boundary between them. So don't sweat the details. That's not very much details yet, but we'll return to this concept again as we read some of our papers this semester. Oh, and here's just for example, it's never as simple as we might make it out, especially in class. This is an example from a paper in 2011 that studies one of the Hox genes, HOXA1, and this is showing all of those relationships between it and genes that it turns on, HNF1B, which I don't know what that is, it's just a bunch of letters and numbers to me at this point. It represses FGFR3, fibroblast growth factor receptor 3. So each of these development genes initiates a cascade, turning on and turning off genes. And the point is that these Hox genes are right smack in the middle of the earliest stages of development. They initiate the instruction, basically. So every Hox gene, whether it's turned on or off, then turns on or off, ultimately, eventually, hundreds of different genes in different cells. And it's that combination that instructs each cell what sort of tissue to become. It's not just the expression of one gene, it's the expression of a whole set of genes, the turning on of lots of different proteins that ultimately create sort of a code that tells a cell what type of cell it's going to be. But then there's the situation of time. So not only does a cell have to know what cell it's supposed to become, but it has to be able to turn on and turn off proteins, genes. And this is just economics. So this one, I think, of all the concepts sort of in basic developmental biology, this one might be the easiest. If you're a cell that has had that set of genes turned on that make you an eye, the transcription factors turning on more transcription factors, and you become an eye, does an eye cell keep producing the proteins that tell it it's an eye? So the answer is no. It's already an eye cell. It's already got its identity. It's already in your eye. It's already created the structures that absorb light and transmit that energy through neurons to your brain to let you see. So the economics here is that at different stages in development, different sets of genes will turn on and then they'll turn off because the cell doesn't need them anymore because it already got its instructions that, you're going to be a muscle cell. Okay, that was great. That basic instruction was given to me. Now I need to figure out what kind of a muscle cell I'm going to be. So you can stop telling me I'm a muscle. I already know that. Because otherwise it's just a waste. Cells are just making extra proteins that they don't need anymore. So what we'll see also then is that not only do genes turn on, but then at some point the organism, it's in its best interest to not have those genes expressed, those proteins produced anymore, because it's just wasting cellular resources. No reason to make those proteins anymore. And one of the ways that happens is, again, going back to some important concepts in the central dogma. How long does DNA last? The molecule itself. Sitting in your cell, say it's a cell that's not dividing anymore. It's just a bone cell. It stays around for a really long time. So DNA doesn't degrade. In our cells, in our nuclei where the DNA sits, it's protected, it gets repaired when damage is done by other proteins in the cell that are meant to repair DNA when it gets damaged. So DNA doesn't really change that much except for when mutations happen, which is pretty rare. RNA, on the other hand, when DNA gets transcribed and produces RNA, depending on what sort of RNA molecule it is really short-lived. Minutes, hours in our cells. And that is affected by proteins in our cells. So there are proteins that are they're actually designed and meant to destroy RNA molecules in our cells. We do it to ourselves because we want to be able to alter the expression of genes over time. So if you think about it another way, if you transcribed a gene and you had an RNA molecule that encoded a protein, if that molecule, if that RNA molecule is there forever, let's say it's HOXA1, early transcription factor, it's meant just to tell you which part of your body's up, your anterior. If that RNA molecule stayed around forever, then the ribosomes would translate it forever, turn it into a protein, and RNA molecules can be read multiple times by the ribosomes. So if this molecule was stable, you would just keep making HOXA1 protein, more protein, more protein, probably at a steady state forever. So we have then proteins in our cells that degrade RNAs, so RNAs don't last very long. If a cell wants to keep making a protein, it has to keep making the RNA to make the protein, and our cells also have proteins that destroy proteins, so that the proteins don't stick around forever. And it's these processes that give the cell ability to change at any point in time what proteins are produced in the cell, because they're constantly being destroyed. They get recycled. So there's some, it's not wasteful. The RNAs and the proteins get recycled into their individual components, nucleotides for RNAs and amino acids for proteins, and then those get turned into different RNAs and different proteins by the cell. So we have to think about genetics and development not only in terms of gene expression, but how it changes over time in any particular cell, because it can. And then we get back to a point you were making, Joey, about how do I wind up sticking to the board in the first place. That's all about protein concentration, which is why it's important to know a little bit about the fact that the cells are constantly destroying proteins. Because, well, if there's just one of me, I already wandered around once, I don't need to do it again because I think I accidentally knocked somebody's laptop charger out of the desk. But if it's just me in the room and I'm trying to interact with the whiteboard, but I don't know what I'm doing because I'm just a molecule and I don't have a brain and I'm just randomly walking around, the chance of me hitting the whiteboard, the DNA molecule, the chance of me binding my target DNA site is low. But what happens when I increase the number of me in the room? Then one of me is more likely to hit the whiteboard. So if there are more transcription factor proteins in the nucleus, they're more and more likely to interact with the sequences that they're meant to bind to, they're designed to bind to. So to put that all then in one diagram, we can have situations of low transcription factor concentration, where on this side of this gradient, this molecule is less frequently bound to DNA. But if you increase the number of molecules, if you increase their concentration of this protein, they're more likely to bump into that binding site and be more frequently bound in the nucleus, bound to DNA, that is. And what controls whether or not you have few molecules of protein or a lot of molecules of protein are how frequently the DNA that encodes this protein is transcribed and translated, that is, how frequently the cell makes new proteins, increases the number of proteins, and that's counteracted by RNAs and proteins being destroyed by the cell. This is a very much still an oversimplification. There are lots of other factors that also influence protein concentration. But these are the two main ones. So cells strike this fine balance between too much protein, not enough protein. Any questions about any of this? Not just that point. But. Okay, so DNA makes proteins. Some proteins are transcription factors. They're responsible for turning on other genes. The set of genes that get turned on specify cell type. And cells can regulate how frequently genes get turned on in many ways, including density or concentration of proteins. Now this, we've already seen once, just to reiterate another introductory point, which is it's not just genetics. Like I just said, there are many ways that the cell gets influenced about what genes to turn on and what genes to turn off, how to develop. And we already saw this cartoon of a hormone binding to this protein. And only when the hormone is bound to receptor protein is receptor protein active as a transcription factor that can go to a cell and turn on other genes. So that's the environment. Right? Whether or not this cell responds has entirely to do with what other cells are telling it to do by sending them hormones from the medulla or some other organ. Not the medulla. What am I thinking? Well, let's say the pituitary gland. Random organs that can excrete hormones. So there are other ways that the environment can influence gene expression. Temperature, if you're an organism that doesn't regulate its own body temperature, right? Temperature causes proteins to unfold. High temperatures do. So protein structure changes with temperature. So if you're an ectotherm, and you're just out in, right now, if you're an insect and you're outside, your proteins are going to be a little bit more unfolded, different shape. Maybe they don't work as effectively. So maybe that influences development. So it's not just genetics that influence development. Temperature often influences development. Depending on where you live, pH can influence your development because that affects how your proteins form, that is, how they're shaped, in other words. So it's not all genetics, but in developmental biology, we're mostly going to focus on genetics. But we have to keep in the back of our minds how is, especially if you look at experimental procedures for the papers we're going to read, mouse study. How are the mouses, how are the mice housed? What temperature was the room at? What were they being fed? If you're comparing two different groups of organisms, do you have to feed them the same way, the same stuff, so they get the same nutrients? Because nutrition also affects development. Oh, and then I added, this is just a summary, I'm not going to read it. I added one other point, I guess, to this slide, which is chromosome structure. So the DNA in the nucleus can be really loose, kind of like cotton candy. It's diffuse. Every single double helix is just like floating around in the nucleus. And then at other times, some parts of the chromosomes can be more compact. Proteins, other proteins, condense the DNA into a structure so tight that no proteins can read it. So the transcription factors can't actually get to the whiteboard. They're blocked because the whiteboard, let's say in the case of chromatin structure, DNA structure, some proteins will come up to the whiteboard and crush it into a little tiny ball, like a big trash compactor. And then I can't get to the whiteboard because I can't find the spot in the whiteboard where my DNA sequence is that I bind to. That's a reversible process. So once the DNA is compacted, it can be uncompacted when it's time for the genes in that part of the whiteboard to be read and transcribed and translated. That's another process that can influence gene expression, thereby development. Okay. So I just want to show another couple of examples. Plenty of time. Last week I mentioned, but I didn't show you because of the projector, the Drosophila apteris mutant. So it's got one mutation in one gene that causes it not to grow wings. Here's Drosophila melanogaster, the fruit fly, mentioned earlier. So given that really brief and really fast introduction, there might be a lot of different ways that you could make a fruit fly that doesn't make wings by tinkering with DNA. So let's say this is a double helix. Diagram just as a single line, so it's a chromosome. And there's a gene here that causes wing development, like apteris. So the arrow here is the common way to indicate where a gene starts on a chromosome, little right angle arrow. So there's a wing gene there. And so how could you break this protein? How could you make it stop signaling the body to develop wings?
Transcription
Hmm?
function? You inhibit the transcription function for that?
Okay, so you could turn off, you could somehow disable the proteins that turn on. So that's regulatory regions. So there could be a specific DNA sequence here, I'm just making it up, that would normally bind a transcription factor. But let's just say you have a mutation that removes one of those nucleotides or changes it to a different letter. Now, this transcription factor doesn't recognize that part of the DNA. It's not attracted to the whiteboard anymore, so it doesn't stick there. So this gene never turns on. So that's what regulatory mutations are. It's not affecting the protein itself, which is the first point. Protein coding regions could evolve. So you could have mutation in the wing gene itself, the part that makes the protein, so that the protein is different and can't function to tell the fly to make wings. You could turn off, you could accomplish the same goal by a mutation that prevents that protein from ever being made in the first place. And we'll talk briefly about this third point, whole genes could evolve. This is big picture here is, there are many ways to break the function of a gene or a protein. There are lots of different ways, especially because there's not just one particular mutation here that would get rid of this transcription factor's DNA sequence that it recognizes. Because it could have mutated that nucleotide and had the same effect, or that base, or that base, or that base. And there are lots of different mutations in a protein, which could be hundreds or thousands of nucleotides, base pairs long. There are lots of different individual mutations, each of which could break the function of that protein and have the same outcome, no wings. So it almost sounds like a desperate situation. How are we ever going to figure out how mutations cause developmental change if there's so many ways to elicit or bring about developmental change? And that's one of the reasons why people think this field is so fascinating, because what we're going to read, and there are many cases beyond those that we're going to read, are ways when scientists actually figured out how development changes. You know, which mutations cause what sort of effect on organisms? And we'll see them both in protein coding regions, that is the proteins themselves, but also in the regulatory regions. Okay, so Hox genes. Quick introduction. There are a lot of different genes that are called Hox genes. And this is an example of a diagram of one chromosome. I think it's chromosome 2 in Drosophila. Each of these genes is found next to each other, next to one another, on the chromosome, which is kind of odd for genes. They're not usually, genes that are involved in the same function aren't always in the same region of the same chromosome. They're usually just spread about all the chromosomes. So it makes the Hox genes a little bit weird. And the weirder thing about them is that if you look at where each of these genes is turned on in the body of the fly, or in other words, what part of the fly it regulates or causes to be developed, the genes at one end of the complex are head genes, and then you move into the genes that are expressed or specify thorax, and then ABD, the genes, the abdomen defining genes. So it's really odd. And genetics is not usually like this. It's not usually so organized on a chromosome, which is interesting in itself. So these genes establish head to tail. So some of these genes specify specific structures in the head, and then as you move across that gene, for example, antennapedia, ANTP, normally produces thorax and wings, but if you accidentally mutate it, then it is actually, phenotype is antennapedia. It's pedia, foot. Its antennae actually turn into legs if you mutate that gene. So these are all really important genes for getting development right, and if you mutate them, really weird things happen. So the thing about any gene that we call a Hox gene is it has one thing in common to other genes, which is it has a particular type of DNA binding domain. So these are DNA binding proteins. They're classical transcription factors, meaning that they bind DNA and regulate gene expression. And as I mentioned, but didn't show you last week, maybe you looked at the slides afterwards, I mentioned that you can have this mutant fruit fly that doesn't have a functioning apterous gene, but you can you can provide it with the human version of that same gene, and then it grows wings again. Again, making the point that these developmental genes are so critical to function of an organism that they never change. The common ancestor of fruit flies and humans, if you can imagine what the common ancestor of fruit flies and humans would look like, had Hox genes that caused anterior-posterior patterning of those organisms. And so Drosophila still uses basically the same protein that humans use to tell the developing embryo where your head is and where your tail is. Although we later lost our tails, but that's a separate Evo-Devo question. So that should make it pretty straightforward to study the evolution of development, right? It's easy. If you want to change development, you just change something about a Hox gene. Like, you mutate the gene UBX. But if you do that, then you grow a second pair of wings. That's really cool, actually. That's Evo-Devo. So there's a Hox gene, we'll call it specifically this one gene of the Hox class of genes. It's called ultrabithorax. This is what happens when you get rid of it. The organism doesn't know what it's doing really when it develops its thorax. So it builds two thoraxes and two sets of wings. That's Evo-Devo. Development has evolved. We've got a developmental change. Voila, the end. Except that that's a pretty big change. And for most organisms, really rapid, large changes are not this is not always true, but most always are not advantageous. So say you build a second thorax with a second set of wings. Does that make you fly better? Not necessarily. Is the second pair of wings functional? Did you change the nervous system at the same time so that the second set of wings gets nerves that also wire to the brain so that the organism can control both sets of wings in coordination with each other so it can fly, right? Development is hard to get right. It's easy to screw up. And so large changes are often not useful. Usually it's very subtle changes that over time make a big difference. And we'll see examples of both. We'll see examples in this class of subtle changes and ones that are really rare, which would be huge sorts of changes that are commonplace, that are immediately useful, evolutionarily advantageous. I'm not going to go through this just because we're going to do, as somebody picked, talking about fly development. So I'm going to leave this for that presentation. So you did it. So it looks like Kevin chose it as first choice. Pigmentation and wing patterning. So this is all about the role of those genes in pigmentation. Okay, one last point, although there are a lot of slides about this, but I'm going to go through them. They're not all necessary. So one way to do Evo-Devo in sort of big picture, large classes, we've got mutations, individual mutations that either change regulatory regions or protein coding regions. The third thing, which I didn't mention a few slides back, is gene duplication. That is, sometimes, by chance, parts of our chromosomes get replicated. So we get more DNA that contains copies of a set of genes. So you might think of this as, let's say there's a part of a chromosome that has the Hox genes in red. Randomly, just by chance, in rare events, maybe spanning tens or hundreds of thousands of years in between occurrences, you might randomly get a chromosome break, make an extra copy of those Hox genes. So you've got two sets of the same cluster of Hox genes on the chromosome. You don't have to take my word for it, but trust me, this happens. And people have studied specifically the Hox genes over time and how many sets of that cluster of Hox genes these different organisms have. So you have this huge variation, which is odd, but so some fish have 4 sets, some fish have 7 or 8 sets of the same genes, these central Hox genes that are critical for development. So the question I'm going to leave you with then, after a couple more slides, is answering the question, how does gene duplication help? How does having extra copies of a gene help? Is it helpful? Is it advantageous or disadvantageous? So we don't need that. So another version of that gene duplication horrible drawing that I made. There's a nicer version, but showing the same thing, Hox duplication. So in 2002, this group looked at different organisms, fruit flies, mice, and zebrafish, which we all heard about at the start of class, and they looked at the Hox gene cluster. So these are Hox genes 1 through 13, and these dots just indicate which genes exist in these different organisms. So zebrafish have 7 different parts of different chromosomes that have those copies of this cluster. But you'll notice that sometimes they've got most, so 1, 2, 3, 4, 5, 6, 7 different regions in their chromosomes have the same set of genes. So there's one part here, Hox 9, 2 copies have been lost. They're not there anymore. They were there initially because this whole duplication occurred. So initially, every chromosome had every one of these genes on it. So if there's a pattern, what's the pattern? Has any one gene, there were initially 7 different copies, 1, 2, 3, 4, 5, 6, 7 of every one of the Hox genes, right? These ones specify head, middle of the body, tail. What's interesting about this, or what's an interesting pattern? What don't you see? Is there any one of the 7 parts of the chromosome that still has all of the Hox genes? All 13? Is there one chromosome that has all 13 of the dots all in the same region? No. Is there any one Hox gene that's been lost from all 7 parts of the genome? So the organism still has one of every Hox gene. It's got Hox 1, Hox 2, Hox 3, Hox 4, Hox 5, Hox 6. It's only got one Hox 7 left, so you better make sure that that one doesn't get mutated, or then you don't have Hox 7. So it's got multiple copies of every Hox gene. So can anybody think of any reason why that might be useful? I mean, is it good or bad to have extra copies of genes?
Is it like a backup if something goes wrong that have a spare?
Absolutely. So if you're an essential development gene, what happens if you get mutated? You go all ultra bithorax, and then you're a human walking around with 4 sets of arms, 2 sets of arms, 4 arms. Right, so that's one hypothesis that's in the literature, is that these are such essential genes that maybe it's a good idea to have lots of copies of them. And maybe that's what's happening here with Hox 7 is, and I didn't explain this very much, but the absence of the little circles here indicating the presence of the gene suggests that mutations, DNA sequence changes, occurred in those spots of those chromosomes so that the gene remnants are still there on those parts of the chromosomes, but the gene isn't functional anymore. So it was there, but now it's been mutated so much by random mutation that happens that it's not a functional gene. So I guess it's a good thing that it had 7 copies because it's still got one good copy left to affect that part of development. Okay, so I'll leave you to look at this if you want to. I'm going to show you the last few slides and then we're going to end. These are the ways that duplicate genes can be useful. There are 3 different types and they have actual names that I'm going to hope you remember and use again in the future. So if you've got one gene, and then all of a sudden, I know this is an old slide, these cell phones don't look like today's cell phones. Actually, they didn't look like cell phones when I made this slide either, or modern cell phones. But if you have two, all of a sudden, you've got a spare one. So you could do something like neo-functionalization, whereas you can use one of these to continue doing the thing that it always did. So let's say you've got a HOX gene, now you've got two HOX1s. Well, this copy of HOX1 could keep being HOX1 that makes your head. Well, now the genome, if I can be a little bit anthropomorphic about the genome and assume that it knows what it's doing, if you're DNA, says, oh, I've got another copy of this gene, I could let mutations happen to it and it might do something new for me. Get a new function, like act as a doorstop. Or take on some new role in the body. That's so duplication of a gene, one copy keeps doing what it's doing, then there's neo-functionalization. And all of these processes, the three of them that I'm going to end with, all have to do with mutation. So in this case, this copy of the gene would have to get some nucleotide sequence changes, DNA sequence changes, that cause it to do something else in the body. And we can't predict at first whether that new thing would be good or bad for the organism. But it's new, be it bad or good. So that's neo-functionalization. And then if your gene that you're messing with does multiple things, if that protein has multiple jobs, then mutations to the duplicate genes could sub-functionalize. That means if this is both a computer and a telephone, it can let you talk to people and you can also look up things on the web. If you've got two copies, well, maybe some mutations happen to this copy that breaks its ability to connect to the internet. But it's still good as a phone, so it still does that job for the organism. And on this trajectory, some mutations arise that break the telephone part. So it still does its job for the organism, but it's its second role. That's sub-functionalization. Had two functions initially, and then the two copies of the genes each keep one of those two functions. And finally, the backup argument, non-functionalization. Two copies of the gene, that gives you one that you can get mutations in, and it doesn't matter to the organism. So it's broken. Who cares? I've still got one functioning cell phone.